All right, good morning, everybody. This is AP Bio Unit 6, Part 2, okay? Part 2, which is DNA replication. There's kind of a lot to this. It gets technical. You're going to hear some new enzymes. But we have some stuff to support you. There's a couple videos on my site to kind of back this up. Some of this is very visual on how it works. And you may have had a little bit of this before in your sophomore biology class, but you probably, this is sort of a beefed up version of it. So you'll probably need to review this, more than just watching this one time. All right, so with DNA replication, this happens during the S phase, okay? The synthesis phase. So everything we're talking about is one phase of the cell cycle, and we're just going to really concentrate on what DNA is doing during that phase. Okay, so let's look at how that happens, okay? In the sort of history of looking for how this happened, which you're going to watch some videos on the actual historical experiments that were done, there were a couple, we came up with the structure, so now we want to know, well, how does it copy itself? Okay, we knew it had to copy because it's in every cell. The first method was called conservative. Okay, there's two sides to DNA, and they thought that it just made a whole new copy. That's what the blue right there is, okay? You have the original remains, and you have an entirely new copy of both sides. The next one, called semi-conservative, means new copy, half is new, half is old. So you conserve, or you hold on to, half of the original, and then the other half is a new copy. All right, and the other way people thought it worked was called dispersive, and that's a little here and a little there. Okay, they thought that the little pieces kind of assembled themselves on their own, kind of like building a new Lego structure. All right, now it turns out the actual model is semi-conservative. So this is the one that we know for a fact is how it works. Okay, so the semi-conservative model is what it's called. So just to kind of go through what these are individually, I don't want to spend too much time on this. They thought in the conservative model, they thought the original was entirely maintained. Okay, that's not how it really works. In the semi-conservative, it was half and half. Half is new, half is old. Okay, so each daughter copy is half the original. And this is how it actually works. And the dispersive model, we thought that random pieces randomly assembled, and you got a new mix of half new and half old. Okay, this is not how it works. So how do we come up with which model is correct? They did this really cool experiment. Meselson and Stahl, this is 1950s. Okay, so this, there are people alive today that were around when this happened. So in this experiment, what we did was we took isotopes of nitrogen. There's a lot of nitrogen in DNA. So they bred this E. coli, and they gave it this heavy nitrogen, okay, and it incorporated that heavy nitrogen into its DNA. And then they allowed it to copy, and they looked at how it worked, and they saw that every generation, the amount of that heavy nitrogen got cut in half. So if you look down here at this little chart, you start with 100%. Then it gets cut in half. Okay? Over here, it gets cut in half, and it just kept getting cut in half over and over again. And what we saw was the regular nitrogen, it's almost like a half-life experiment, because half of it is new every time. And that was the evidence for this semi-conservative model. So they didn't directly observe it, they used indirect observation to come up with this. So they grew bacteria with an isotope in it to keep track of where things were going. After they grew it, they put it into a nutrients with regular nitrogen, the light regular isotope of nitrogen. All right. Bacteria are great to do these kinds of experiments on because they breed very, very quickly. And so we just kept seeing the heavy nitrogen get cut in half with each generation. And that gave us the evidence for what we call the semi-conservative model. And you can see you can get 4 generations of E. coli in 80 minutes. As long as you keep it nice and warm and feed it, it grows very, very quickly until it runs out of space. Okay. So here's just a diagram kind of showing you go from 100% heavy nitrogen, 50-50, 25, 20. So it just keeps getting cut down every single time. Okay. So that's how we came up with the evidence that this is how it works. Okay. Now additional work has been done in this area to give us more and more details. So we're going to look at what's actually happening here. Okay. So let's take a look here. DNA copying begins at sites called origins of replication. So where's it going to start? Now, if you remember in our last video, I talked about the 5 prime and the 3 prime end, and how those are named for the points on the carbon rings of the ribose sugars. Okay. And so we have this 3 and 5 end, and they have to go in a particular direction we're going to talk about here. So when this happens, lots of different proteins are attaching to these points, and they unzip or open the DNA to give a replication fork. So think of this as like a fork in the road where the road splits. DNA is got a fork right here, okay? So they call this the replication fork. Okay? Replication fork. And that's where all this happens. All right, there is a Cami out there if you want to kind of follow along. I have copies of the packet for this unit coming. Should be done today. They're not, the packets are 90 pages, so the copy center was going to make them into books, but you can't make books that big. So if you want one, it'll be here the next time you meet. All right. There's your replication fork. So this happens in multiple steps, okay? Let's go over these steps. You should be familiar with what enzymes and what and where things are happening. Okay. First thing we have is an enzyme called helicase. Okay? Helicase unzips DNA. There's lots of biology jokes about unzipping your genes and helicase. Okay, you can write your own jokes. So helicase unzips DNA. Well, what does that mean? It splits the two sides, the two backbones, and it makes the fork. Okay, DNA wants to bond with itself. These base pairs want to remake those hydrogen bonds. Okay, to stop that from happening, there are proteins that sort of brace the DNA open. Okay, it's kind of like holding the DNA open. Those are called single strand binding proteins. Guess what they do? They bind to single strands of DNA. So this is one of those, students like these kinds of names because the name describes what it does. Topoisomerase. Okay? This stops DNA from coiling back up into basically a chromosome-like structure. We don't want that DNA to twist back up. We have to have that section unzipped, and then we have to have it held in place so that this can happen. All right. Yeah, go back. You got it. I thought that might have been a little too fast. People at home, you guys with me? Can you just kind of check in and say, yep, I'm here, in a chat? Justin, thank you. Looking for everybody out there. Go where? You just want me to talk about it again? Okay, so that particular enzyme stops the... Got to remember, DNA is wanting to wind up in the chromosomes, okay? And so if we kind of like zoom in on one little section, we have to hold this section open so it doesn't spring up into that, okay? I'm trying to think of something that might be springy that you kind of have to hold in place, that you could think of it like, if you had a big slinky, and you were maybe like working on just one little spot in it, you'd have to hold the slinky stretched open, okay? Slinky's not the best analogy because it's just one coil. DNA is actually sort of two coils. And they call this structure over here supercoiling. All right. All right, back to this picture. I kind of just drew this. This is uncoiled DNA, sort of down at the basic level. All right, here comes helicase. What does it do again? Unzips. Unzips. All right. These are the single side binding proteins. What do? And their purpose though is to hold it open, hold things in place. Remember, it wants to zip back up. Even though helicase is coming in and unzipping, it wants to collapse right back together. Okay, these bonds between these bases, they want to reconnect. chemically. Okay? Topoisomerase holds the DNA and stops it from supercoiling back up. Like this whole thing wants to wind up on itself. Okay? This enzyme stops that from happening. So you've got 3 things so far you need to know. You need to know what they do, what they're called. All right. There are more coming. So this is just the early stages. All right. Now we have an enzyme called primase. Okay? Has anyone ever worked with swimming pool pumps that won't start until you prime them? Has anyone ever heard of that sort of thing? All right, I knew you would have. So to prime something, if someone says, yeah, we've got to prime the pump, sometimes you have to pour some water in a pump to get it started. So when they say prime it, this is not like prime numbers or things like that. This means starter. That's what that means. Okay, and that's what initiate means, start, okay? So the enzyme primase starts replication by adding short sections of RNA, and they're going to call them primers. That's kind of nice. So primase adds primers. All right? Why do we need this? It turns out that DNA can only attach to places where there are existing nucleotides. It doesn't like to just, let's say this is one side right here. It doesn't like to just randomly attach nucleotides in random spots, okay? It can't do that. It has to basically do it like this. Okay, you have a strand of DNA. I'm going to draw the primase in red. Well, that'd be the primer. Primase puts that in place. Now it will go like this. It'll go in order. Okay, it likes to stay in order. And it builds one at a time in order. It can't just randomly assemble itself. Every time it does that, the backbone of the sugar and phosphate make a bond, and the bases make a bond. Okay, so those reactions have to happen in order. But when I say reactions, I mean bonding. All right. Erase all this. I bet there's a quick erase command, and I don't know what it is. All right. So primers are the starters or the foundations or the cornerstones. Primer means to start, like we talked about. So let's take a look at what happens here. The primer comes in. Okay, it's RNA, so it's not going to stay there. Okay, when you copy DNA, you're not going to add all this random RNA in here. Otherwise, every copy would start filling up with RNA. Okay. Antiparallel elongation. Okay? What that means is, here's my fork. One side is going to grow in this direction, and the other side is going to grow in that direction. Okay? So the direction they grow is actually very, very important. Antiparallel elongation. Oh, I found the clear all button. Okay, a lot going on here. if I'm going to make a polymer is a long molecule. We studied them at the beginning of the course. DNA polymerase polymerizes DNA. It makes little nucleotides into big strands of DNA. This always happens in the 3 to 5 direction. Okay? It moves in the 3 to 5 direction. The new ones that are laid down are going in the opposite direction. So the new ones are always in an opposite configuration as the originals, okay? So one strand of DNA goes 5 to 3, okay? And it's going to move this way. That's the direction it builds. It builds 5 to 3, but when it lays them down, it's going to lay them down this way. Remember, DNA is antiparallel. One side goes one way, the other side goes the other way. Start thinking of 3s and 5s as like starts and finishes, okay? It's almost like if I was going to build Lego blocks, if I took Lego blocks, I would stack them up always the same way on one side, and then on the other side, they would build upside down, okay? Lego blocks only go together one way. You can't put the two little studs on top and like smash them together, okay? So if we're working with very basic original Lego blocks, we just keep building, 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 and the little bump on the top of a Lego block goes into the hole on the bottom, okay? That's sort of that 5 to 3 on one side and 3 to 5 on the other side, okay? When it builds, it goes in the 3 to 5 direction. All right, so DNA polymerase follows Helicase on what's called the leading strand, okay? The leading strand kind of like just keeps building out of the fork. The lagging strand has to go the opposite way, and it needs primers over and over and over to get it started. This is very hard to understand without watching an animation of it. And that's what we're going to do in a little bit at the end of this lecture. So one of them, once it starts, it just keeps going. The other one has to work backwards, so it has to keep getting started. Okay. This is the leading strand up here on the top. As this cuts, it will just keep going, okay? It goes this way. This one has to have starters to keep filling in the gaps. It has to keep filling in the gaps because it goes the other way. It constantly needs starters. What do we call these little starters? Primers. So the leading strand only needs one primer to get started, and then it just keeps going, okay? If you're going this way, you only need one starting spot, okay? If you're going this way, you have to keep putting starters in here. You have to keep putting starters in here. All right, there's your DNA polymerase jumping on your primer, and like I said, it goes toward the fork. The lagging strand goes away from the fork. This is complex. There's a lot of little nuanced points to it. You're probably going to have to review this more than once. All right. So that leading strand just keeps making one continuous strip. The lagging strand has to constantly have a new starting point, and it works in chunks. So the leading strand just keeps going. The lagging strand kind of has to come in chunks. Once you see the video, this will make sense. I'll try to put the video in the YouTube description. It's something I normally don't do, but I think it's very important that you watch these other videos. So, Okazaki fragments are the name of these little chunks that come in on the lagging strand. So know that if you're going to connect this word to something, it should be lagging. Okay? Okazaki, lagging. Now, this is called an eponym. In science, we are trying to get away from these, okay, where we name things after people, especially in anatomy. Some of them have been around and they're so accepted it'll probably be hard to get rid of them. All right, so did you see that real quick? Watch that animation again. This is DNA polymerase. It's going to sort of follow the leader. The leader is helicase, okay? It's just going to keep going. This is going to keep unzipping right here, okay? So this will keep unzipping in this direction. So that's going to split apart, and then this will be following it, and it will just keep building. Okay, watch the little animation come in. And you saw it kind of zip away. On the bottom, it has to start on an individual primer every time and bring in these little things called Okazaki fragments to kind of build in the opposite direction. Okay, after DNA polymerase makes these fragments, it's going to take the RNA, the RNA primers. Those are going to come out. And when they come out, they'll be replaced with DNA, because we're going to match DNA with DNA. You don't want to have DNA with little chunks of RNA in it. They do build together, and we'll talk more about that later when we talk about protein synthesis, but we have to remove those because we want to keep DNA. Well, you got to take the RNA that was just sort of a tool to build it out. All right. Ligase is another enzyme that joins these fragments together. Okay? If you ligate something in surgery, you tie a knot in it. Okay? So this is sort of like tying off these little fragments. There was a surgery where women would get their fallopian tubes tied, and some people will say, did you get your tubes tied, like if you don't want to have any more babies? The medical name for that is a tubal ligation. So they tie a little string around your fallopian tubes, and that closes them off. No more sperm can get in, no more babies. Sort of a permanent birth control. All right, let's take a look at this again. There's your DNA polymerase. What are those doing? They're taking out the primers and replacing them. And ligase connects things together. Okay, it's kind of like the opposite of helicase. Helicase splits them apart, ligase connects them together. So remember, ligate, tie together. All right, there are a couple YouTube videos at this point. I want to stop my lecture and have you guys go watch those. Give me 1 second here. I don't know if I want to stop exactly. All right, let me add one or two little things, and then we'll go watch the video. I don't want to end my video just yet. Okay, there are some special problems that happen at the 5 end. Okay, remember the 5 end is going to sort of be the free end. Okay, so what happens here? DNA polymerase can only add nucleotides to a 3 end. Okay? So there has to be an end of DNA that goes the other way, and that's going to be the 5 end of the lagging strand. Okay? There are special structures called telomeres that are on the end of nucleotides. They do not code for genes. Okay? They make a little cap on the end of DNA. Part of aging is these getting shorter and shorter and shorter. As you age, these get short. And keeping these maintained, people think might be a way to fight aging. Okay? Aging is multifactorial. It's not just, well, if we stop this, you'll stay young forever. That's not really how it works, but it will help. As you get older, these get shorter to the point where the ends of your genes start eroding. So we can tell how old a strand of DNA is by looking at how much of the telomere is left. There's an enzyme called telomerase that adds telomeres back to the ends of DNA. Okay, it's almost like, tying a knot in the end of a rope so it doesn't go through a hole or something like that. That's kind of what these are. They get shorter with age. All right, there's a lot here, so I'll kind of take it slow so you can get caught up. We're almost done. Turn you into those short videos, which are very important to understand this. Like, you don't really get a picture of how it works in your head until you see a really nice animation of it. This happens very, very quickly. DNA can copy very, very fast. I'm watching my in-class note takers, just kind of letting them be my cue. All right, we feeling okay over there? Good. See a thumbs up. All right. One last thing. During DNA replication, mistakes happen. Okay? So there are mechanisms that come in, proofread it, and fix the mistakes. Okay? So DNA polymerase adds, it also proofreads the bases. Okay? There are enzymes, and we're not going to get into the particular names of them in this course, that will come in and fix mismatched nucleotides. So let's say you're going through, you got an A and a T, those match up, and then you've got an A and a G. Okay? That doesn't match. So an enzyme will come in, take out the mismatched one, and fix it. it's a multi-enzyme process, but one of them is called nuclease. All right, this is a heavy vocabulary unit. All right, so you really might want to make some flip cards to keep track of things. You're probably going to have to review this more than once. Going through maybe and watching this video again, I try to underline all these new terms. Maybe watch somebody else's video. All right, I'm going to go ahead and stop here. I'm going to turn you guys loose to do these 2 short little ed puzzles. And I'd like those done in class. People at home, you guys can sort of disconnect and get those done. I'll post this YouTube as soon as I get it rendered.